Hello, everyone, and welcome to today's webinar, The Value of ECAs for Regulatory and HTA Evidence. Today's webinar is brought to you by ISPOR corporate partner, Genesis Research Group. Before we begin, I'd like to review some housekeeping reminders. All attendees have joined in listen-only mode. Please submit your questions to our speakers using the Q&A feature. Our speakers are looking forward to answering your questions after the presentations. Finally, the webinar is being recorded and will be available for on-demand viewing via email and on the ISPOR webinar series webpage in approximately 2 business days. With this, I would like to turn it over to today's moderator to begin.
Thank you, Jess. We're thrilled that you've joined us to discuss the value of external control arms, or ECAs, for regulatory and HTA evidence needs. My name is Alexandra Sosinski, and I'll be today's moderator. I'm a Senior Director of Pharmacoepidemiology and Safety at Genesis Research Group. I'm joined by Mira Pavlovic and Frank Ulrich Frick. Mira is a medical doctor in dermatology and an expert in regulatory and health technology assessment in Europe, based in Paris, France. Frank Ulrich is an expert health economist, academic dean, and a member of the Arbitration Board on Drug Prices, based in Nuremberg, Germany. During today's webinar, we will share insights on how to strategically navigate ECA investment, acceptance, and policy to maximize impact and return on investment in regulatory and HTA decision-making. I'll be kicking off today's webinar with a presentation on advances in addressing ECA shortcomings to boost return on investment. To set the stage, ECAs are comparison groups designed to closely align with the trial population. ECAs are often derived using patient-level data from disease registries or electronic health records, and they are used to augment or in some cases replace a traditional control arm when randomization isn't feasible. ECAs can be historical or concurrent, or contain treated or untreated patients. ECAs are gaining traction for regulatory and HTA evidence needs, particularly in oncology and rare diseases when randomization is not ethical or feasible, but their true value and acceptability in practice is debated. Now that we've outlined what ECAs are, let's explore the essential building blocks that help determine their success in practice. In this visual, I highlight the 4 cornerstones of an external control study. We'll take a closer look at each of these elements, discussing what good looks like, where there are remaining challenges and pitfalls, and how recent advances are helping to move the field forward. Let's start with stakeholder alignment. Success in this area depends on sponsors leveraging available guidance and actively working with key stakeholders to align on expectations and acceptance criteria. Historically, the lack of clear expectations and precedent outside of RCTs has made ECAs challenging to justify, especially outside of oncology. However, we're seeing progress in this area. FDA released draft guidance outlining considerations for the design and conduct of externally controlled trials in 2023, with final guidance forthcoming. This document from FDA gives sponsors insights into the agency's current thinking, including threats to study validity from potential biases, ensuring data access for the agency, and best practices for communication with FDA. A few months ago, the UK's MHRA has issued a draft guideline on using ECAs based on real-world data to support regulatory decisions. The MHRA draft guideline focuses on the need for thoughtful justification for ECAs, real-world data quality, pre-specification, and careful bias mitigation. The EMA is also working on its own guidance, currently in drafting, further reinforcing regulatory interest and a clear sign that stakeholder alignment is gaining momentum globally. Outside of formal regulatory guidance, there are a number of helpful tools such as SURF, a screening tool published by Dr. Olka Campbell and colleagues in 2023. SURF is a structured way for sponsors to assess whether real-world data might support an effectiveness claim in an FDA application. Criteria to assess for regulatory feasibility include things such as whether there is unmet need and if the natural history of the indication is well established. This tool helps bridge the gap between regulatory guidance and practical needs of sponsors. Lastly, and specifically for select rare diseases, the FDA has just last month introduced new evidence principles designed to facilitate innovative approaches and make regulatory pathways more predictable. Per the RDEP, which was developed jointly by CDER and CBER, sponsors are eligible to apply prior to the launch of a pivotal trial. If using this pathway, one adequate and well-controlled study plus robust confirmatory evidence may be grounds for approval. Confirmatory evidence could include real-world data, and this directly addresses an area where ECAs have the potential to make a big impact. Taken together, these developments are helping to clarify the criteria for success, making stakeholder expectations clearer, and encouraging the thoughtful use of ECAs. Moving now to the second cornerstone, fit-for-purpose data. The data we use for ECAs must be high quality, complete, and reliable for the specific study question and evidence needs. When they arise, operational challenges around data access and privacy have been particularly difficult to overcome. Similarly, missing or inconsistent data can reduce the feasibility of an ECA. Luckily, on this front, there have been several advances. Federated data networks like Darwin, highlighted in the EU's real-world evidence framework, and Sentinel in the US, allow researchers to analyze data from multiple sources without moving or sharing raw patient data. This approach helps address privacy concerns while still enabling robust evidence generation. For ECAs specifically, a federated approach can also result in enhanced sample size and generalizability. Next, tokenization is creating new possibilities for linking patient data across trials and real-world datasets. By using secure tokens to connect records, we can build more complete patient profiles without compromising confidentiality, and this is allowing for richer, more comparable analyses for ECAs. The use of AI and machine learning tools are now being harnessed to curate data at scale by extracting large volumes of complex outcomes, such as cancer progression in this featured large language model example from the team at Flatiron Health. These capabilities are helping to expand beyond traditional data curation methods. And finally, we're seeing a real movement towards democratization of data access, with more platforms and collaborative initiatives making data available to researchers, patient communities, and industry partners alike. Here, I'd like to highlight the Cystic Fibrosis Foundation's sponsor-agnostic shared control repository that enables multiple sponsors to progress pipeline products in parallel. This innovative strategy increases transparency, standardization, and encourages new ways for thinking about external control arm data strategy, particularly for rare diseases or when longer follow-up time is needed. Together, these advances are making it possible for sponsors and investigators to obtain the fit-for-purpose data they need, ultimately supporting more credible, reliable, and impactful ECA studies. The third cornerstone is methodology. It's vital to address confounding factors and other biases through careful study design and analytical choices. Robust research methodology sits at the heart of successful ECAs, but it's also where many big criticisms can arise. Key issues include comparability between patient populations, when and how outcomes are measured, and unmeasured or residual confounding. We're seeing many meaningful methodology advances across several fronts. First, rigorous frameworks like estimands and target trial emulation are now being applied to ECAs. By clearly defining the treatment effect of interest and using trial emulation practices, we can design our external control arm analyses to better mirror the trial and support causal inference. Index date selection is another area of focus. Pinpointing the correct time for cohort entry, or time zero, is critical for ensuring comparability and generating unbiased treatment effect estimates. Colleagues at Genesis, led by Dr. Erin Crowley, have explored how techniques like propensity score matching can help improve index date selection in real-world data from oncology EHRs, where patients may be eligible for inclusion at multiple time points. We're also seeing progress in the analysis of continuous outcomes for ECAs derived from real-world data. Comparability challenges arise when outcome ascertainment occurs at regular and more frequent intervals in the trial versus according to routine clinical care in the ECA. Recent work from Dr. Joan Van Wang at Genesis, presented at this year's joint statistical meeting, proposes a novel framework integrating spline modeling, inverse probability of treatment weighting, and imputation using a Bayesian approach. Key strengths include bias reduction and accounting for uncertainty from both missing data and propensity score estimation. And finally, on the topic of methodology, I would like to highlight the importance of sensitivity analyses and quantitative bias assessment tools as a critical practice in ECAs. As an example, in collaboration with the Causal Lab at Harvard, Dr. Gupta and team recently published a QBA for multiple advanced non-small cell lung cancer trials in which they evaluated the impact of missing data, such as with ECOG performance status. These methods allow us to evaluate the robustness of our findings and quantify the potential impact of residual confounding and other biases, leading to more transparent and rigorous evidence. These methodological advances touch on some of the most pressing challenges that have previously limited the utility of ECAs, and they're helping pave the way for additional innovation. Moving to the last cornerstone, transparency. Here, good looks like sharing how data were selected, as well as study design and analysis decisions to ensure maximum credibility and trust. Recently, there have been important advances to support transparency in every phase of ECA work. Pre-registration in forums such as the RWE registry or the HMA-EMA catalog helps ensure that study designs are finalized in advance, promoting accountability and sharing of methods. Protocol templates like Harper standardize communication of research methods to improve rigor and reproducibility. Data and code sharing initiatives allow others to verify or build upon findings, while structured guidelines such as the target statement and the record PE checklist set clear expectations for the level of detail and transparency required when communicating emulated trials and study findings. When adopted, these tools and practices help ECA researchers achieve the highest standards of scientific integrity, and the badges are fun to show off at conferences too. So ECAs have the strongest chance of being accepted in cases of rare diseases or high unmet need where traditional RCTs are infeasible or unethical. Acceptance is higher with high quality, fit for purpose data, a well-matched external control population, comparable endpoints, and a robust design and statistical methods. Early and ongoing engagement with regulators also goes a long way towards building confidence and clarifying the criteria for acceptance. On the contrary, ECAs are least likely to succeed when a standard RCT is feasible or when there are gaps in data quality, major differences between groups, or uncontrolled bias. If data selection isn't transparent or stakeholders aren't engaged, likelihood of acceptance can also drop. In short, ECAs work best when they fill a necessary gap in evidence, are rigorously designed, and are paired with clear early communication with stakeholders. As you think about pursuing an ECA, remember that success depends not only on whether you can feasibly use one, but also whether you've proactively addressed all four cornerstones we've discussed today to overcome the most common criticisms and challenges. ECAs play a critical role when we need robust comparative evidence, but can't feasibly run a randomized trial. They're especially helpful for benchmarking single-arm trials, using real-world data to contextualize findings, and enhancing the generalizability of trial results. ECAs are also used to compare results to an updated or local standard of care and inform internal decision-making. Ultimately, the strongest justification for an ECA is when you need comparative evidence and there's no alternative. When considering whether to invest in an ECA and its return on investment, it's helpful to consider how each of the cornerstones we discussed impacts costs and potential payoffs. On the investment side, each cornerstone translates into specific resource needs, engaging early and often with stakeholders to align expectations, leveraging high-quality fit-for-purpose data, which could involve innovative access models and/or tech-enabled data curation methods, applying robust research methodology such as QBA, and maintaining transparency starting with pre-registration through clear and detailed reporting. Each of these steps requires purposeful investment in expert staff, data access and curation, methodological and analytical rigor, and stakeholder engagement. As for potential payoffs, when these elements are well done, ECAs may facilitate accelerated approvals, reduce the need for large or multiple randomized trials, and provide strong contextual evidence, especially for single-arm trials. Most importantly, ECAs can help boost confidence in the overall evidence package. To me, success from investment in an ECA ultimately looks like meeting the goal of patients gaining quicker access to much-needed therapies, and a deliberate assessment of return on investment can help ensure that we're making the smartest choices in pursuit of this goal. As we wrap up this first segment of the webinar, it's hopefully clear that ECAs can play an important role in drug development, especially as real-world data becomes more accessible and accepted. ECAs offer key benefits like enhancing the generalizability of trials, allowing for tailored comparisons to local standards of care, and helping to strengthen positioning in a competitive market. However, it's important to remember that ECAs are not suitable for every situation. They're generally used as an exception to the gold standard of an RCT and require strong alignment with stakeholders when deviating from that standard. Identifying and accessing fit-for-purpose data can also be a significant hurdle. To maximize the success and return on investment of ECA studies, a thoughtful approach to all four cornerstones, stakeholder alignment, fit-for-purpose data, methodology, and transparency must be in place. Recent advances, like ECA guidance from regulators, AIML for data curation, novel method approaches to index date selection, and new tools to promote research transparency are helping us design more robust studies that hold up under scrutiny. With these tools, we're better equipped to answer challenging research questions and deliver evidence that matters. Thanks so much for your attention. I'll now pass the floor to Mira to continue today's conversation.
Hello, good morning and good afternoon to everybody. I hope you can hear me well. So my name is Mira Pavlovic. I worked for many years in French agency, then in European Medicine Agency. Then I was one of the directors of HTA. Then I was one of coordinators of European HTA. And I will try to share with you my experience in assessment and reimbursement of external controls. This is the FDA definition, probably you're aware about. So external controls can be either concurrent or historical ones. So concurrent external controls are very rarely seen because it means that subject level data should be collected at the same time as the treatment arm, but in another setting. In this situation, very frequently, companies are asked to do a comparative randomized trial and not a concurrent external controls, but it may happen. Now, with regards to non-concurrent, it is most frequent situation, meaning either you will look for retrospectively collected natural history to see the evolution of the disease and how it manifests, published literature data, previous clinical study in the same setting, or patient as his own control. So this is all called the historical controls. With regards to external controls as compared to a randomized clinical trial, it is just to remind you that RCT is here to distinguish the effect of a new drug from other possible influences. For example, vaccine and waning disease, spontaneous changes in the course, placebo effect, or a kind of biased observation. So what does it mean? It means that if the natural history of a disease is well defined and known, and if we are sure that the disease will not improve in the absence of an intervention, historical controls or external control could be acceptable if randomized control trial is not possible. So we'll see in which situation this can happen. Let's look at this together. So now this slide is 30 years of practice. One, possibility to perform RCT is a key. And this is not my cursor here, so I don't know what happens. Anyway, possibility to perform RCT is a key. If we have rare disease, regulatory and HTA bodies distinguish broadly real orphans, meaning very small population, and a kind of false orphans when they consider that RCT is feasible. So if we are to summarize in one sentence, it's better to have bad RCT than no RCT. And always versus best standard of care. Of course, in a well-defined population, of course, with clinically relevant improvement that should be shown in key symptoms and signs of a disease, and as objective as possible, meaning that surrogate endpoints are not accepted. And meaning also that hard or final objective endpoints are requested whenever possible. Sometimes that they could be replaced by clinically relevant intermediate endpoint because it's also clinically relevant. So once said that, let's see how regulators and HTAs reason. In practice, what do we need to have if we wish that our external control trial is accepted? One, the most important is well-known evolution of a disease. This is the most important. If you have a disease like lupus coming and waning, etc., it's not possible. Of course, well-defined population, a high unmet medical need, and objective and measurable effect, even short-term. Of course, no adequate comparator or no adequate standard of care, and obvious and huge added value as compared to no treatment or to historical controls. Possibility to cure, improve daily living, survival. What do you mean? We mean that if you have a drug that somebody in a wheelchair will take and then stand up and walk, this is okay. If you can cure a disease as Gleevec 20, 30 years ago, it is okay. So it means a huge effect size. So when you look to the FDA guidelines and comments, when you look to this, you will see that the reasoning in regulatory arena is more or less similar. So there is no available therapy. The disease progression is understood and predictable. This is important. outcome measure is objective. Treatment effect is very large. It is measured in a manner that reasonably manages minimizing bias, etc. And more or less that's that. So if you look to the refusals from the EMA, you will find the same reasoning. Randomized control trial was possible, so this is not acceptable. There is no comparator group, and comparator does exist. Randomized control trial was considered feasible. Many risks of bias, so selection bias, etc., so not accepted. Similarity with existing marketing agent, so it means that control trial might have been done, maybe. No dramatic activity, meaning no big effect of a drug, etc., etc. So when you look to that, and when you read FDA guidelines, sorry, it's more or less the same reasoning for regulatory purposes. Now, what about HTA? So comparative or relative effectiveness assessment is a rule. It means that it should be comparative. So that's it. So it is based on direct comparisons coming from double-blind randomized trials. Indirect comparisons are there just to support the efficacy, but should not change or replace direct comparisons. Many HTA bodies do not take into account the nature of marketing authorization, meaning that marketing authorization under exceptional circumstances or a conditional one, they will assess the same thing, meaning evidence. Evidence, and then evidence, and then evidence, and that's it. So in some situations, non-randomized data is accepted. For example, when you have gene therapy, I don't think it's possible to do randomized trial. When you have very invasive procedures, if you can imagine a mock surgery or whatever, in very rare diseases, and when a disease progression is too long to allow for a randomized trial. So there are many papers on that. Replacing RCTs, ECAs, and BAT says it's a rather a choice than a necessity. Don't think it tells you everything. I think Frank Ulrich will tell you more about that. In-house, where I worked, there is a huge a box with all methodological points to take into account when you are thinking about external controls. So it means better not do that. And in Europe, when you look for reimbursement decisions, there are many differences based on the same data. So why did I take this? This is a very old example, but the reasoning didn't change. And I will show you the reasoning 10 years ago and 2 years ago. So it is in my field. It's in metastatic locally advanced basal cell carcinoma, where you cannot do surgery, and it is too big for radiotherapy. There are two drugs. So Vismodegib is 10 years ago or more, and Sonidegib, that's a little bit more recent, where you have a randomized phase 2 study, but it was low dose versus high dose. And only low dose was then approved by EMA. So what happened to this one? So this is about cellular carcinoma, and some photos are before and after. So the design was terrible. I mean, it was an open trial. It was even not shown that if you do a treatment and then you do a surgery, it can cure the patient. It was not done. The only endpoints was overall response rate, and in some patients, progression-free survival, because this is a long disease. It's a huge morbidity and low mortality. And so in New England, it was a great advanced, it's magnifique, it's perfect, because nothing exists. You can do nothing with these patients. So EMA said, okay, because in at least half of patients you have improvement, and that it was not so badly tolerated, so it was approved. And of course, NICE said no, and we shall see why. So what happens with the two? At the end of the day, they were reimbursed in France, for example, and in Germany, because even if data was bad, the quality was bad, it was taken into account that it was a very rare disease, it is severe, it's long evolution, but there is nothing. Radiotherapy is possible, but not on the half of the head. And even if the place in therapeutic strategy was not defined in the trial, people decided that it could be neoadjuvant, meaning shrink the tumor and allow for surgery, and it could be curative in some situations and palliative in others. So it was similar in Germany and in France. And the same drugs were not recommended in UK. It was not recommended in Sweden, for example, because there was no RCT, but there was no direct comparative data. In the case of Sonidegib, comparison to Vismodegib was not accepted. There were no registries. There were no data on overall survival, and they were too expensive, so not cost effective. And the latest decision on those Vismodegib by TLV, it was 2 years ago. So it means that the reasoning for not accepting didn't change. So you know that from January we have mandatory assessment of oncology and advanced therapy drugs in Europe. So there is a common assessment, and member states will remain responsible for conclusions on added value and for taking decisions on pricing and reimbursement. So as you probably know, from January it's all new oncology, advanced therapies, and then there are also some new therapeutic indications for oncology drugs, etc., etc. Okay, so what happens with this? Here you have drugs that are assessed now. These are joint clinical reports from European Commission HTA, and as you see here, you have most anti-oncology drugs. You have one anti-AP, and I'm sure that, for example, for ATMP, I don't see how they can do randomized control, but let's see. So I think it's very important that we see in 6 months how these drugs were reimbursed and what how data were interpreted by European HTA for such situations. But because in many of these drugs, you can imagine external controls. So let's see what happens, and let's analyze in 6 months to 1 year, and we shall know more about this. So that's why I think this should be followed closely, because common clinical evidence will be and is available with these mandatory joint clinical reports. But I think still that common assessment will lead to different reimbursement decisions. So let's follow this. I think this is my last slide. So thank you very much, and I'm open to your questions. Thank you.
So I would like to switch my camera on, but it's blocked by the host. So if you could help me with that. Nope. So hello, everybody. My name is Frank-Ulrich Fricke. I'm an economist by training, and now you should be able to see me. I'm here today most likely because I'm sitting on the arbitration board on drug prices in the German healthcare system for the last 14 years. And I wanted to use my time here during the presentations to give you the flavor of the German approach, basically. So first of all, you are most probably familiar with the German assessment approach, but still, just as a reminder, the first 6 months are a clinical assessment, and based on that clinical assessment, within the second 6 months, there are reimbursement price negotiations with the statutory health insurance. And then if the parties cannot agree, then finally there will be an arbitration. Now, more important than that, the slide I did not include, and that is reimbursement and coverage is given with the EMA label. So the clinical assessment is not meant to decide on reimbursement and coverage. It's meant to prepare the parties for the reimbursement price negotiations, and I think that is most important. Now, the next reminder here is, and I think I can keep that short because you have seen that in previous presentations already. The whole assessment relies on comparative data, sufficient trial duration, and patient-relevant endpoints. And frankly, you need to have it all. Otherwise, you will have a hard time in reimbursement price negotiations, at least in many cases. And next, we are looking into external control arms as basically indirect treatment comparisons. I'm simply repeating what you saw already, so I can very quickly jump over that. I think the most important message here is from a German systems perspective, and I guess that's shared by many of you out there, external control arms are part of an indirect comparison. And when it comes to indirect comparisons, then there is a section on that within the general methods that are used for assessing evidence within the clinical assessment as a preparatory act of the reimbursement price negotiations and prior to the final resolution of the GBA on the additional benefit. So first of all, routine application is not advisable. I highlighted that. And if you go for indirect comparisons, adequate justification would be required. And within the methods, there is an emphasis on unsolved methodological problems. And when it comes to methods for indirect comparisons, then the focus is mainly on adjusted indirect comparisons, as you can take from the slide, as well as network meta-analysis. So that's basically what needs to be borne in mind when it comes to external control arms. Now, prior to my talk here today, I briefly looked into where are we, and I came across that systematic review by Leo et al. 2025, just published in JAMA. And what you can see is their conclusions here, which would create and support concerns on external control arms within an assessment. And I tried to highlight the main aspects of criticism here. And justification is missing. As you can see, pre-specified external controls not always done, confounding not sufficiently taken into account, sensitivity analysis missing, and the complete absence of quantitative bias analysis as well. So these concerns would clearly not support taking into consideration external control arms. Now, when would external controls be acceptable? So first of all, once again, RCTs are the gold standard. I don't think I need to repeat that and spend a lot of time on that one. Single arm trials may require external controls, and here you can see deliberations on the situations where we would look into that. As you can see, not only rare diseases, but extremely rare diseases. And we can discuss what is the difference between rare diseases and extremely rare diseases, and then certain disease constellations where you can take that into account. And I have a couple of examples where we looked into that. And what is important as well, and Mira elaborated on that already, the expected course of the disease is more or less determinate, and hence you could use historical data for comparative purposes. Now, when it comes to accepting or using external controls, then you need huge effect sizes, and that is what we in the German setting call dramatic effects. And again, the prerequisites for a dramatic effect are described in the general methods as well. So deterministic course of the disease, missing treatment options, and then a dramatic effect might be sufficient to assign an additional benefit. Question is, what is a dramatic effect? And I see the highlights are a little bit in different places here, but for that classification, one important aspect is the fateful cause of the disease. That is important. Second, the observed effect cannot be explained simply by confounding factors, a higher significance level of 1%, and then it's about what is the magnitude of the effect, and here the discussion is on an expected relative risk of 10, and then finally, the observed relative risk is a little bit, let's say, ranges put out there, 5 to 10 would be acceptable depending on the various courses. And perhaps most important is that all this can be found in the general methods of the IQWiG that would be applied within the assessment and would be the basis for discussing the additional benefit within the GBA. Now, based on that, let me come up with examples. So one example is here, LibMaldi in MLD, and here you can see even a major additional benefit was assigned based on, as you can take from the slide, I would say, a variety of data sets. Now, having that said, what were the reasons to accept these data as a basis for a major additional benefit? And these were the large effects in morbidity, not explainable by random effects. That was important. And second, the arms used for comparative purposes were sufficiently structurally similar so that a quantification of the additional benefit was possible even without randomization. So that's my first example. And if you want to look that up, these are assessments that are available at the website of the GBA, and they are in English. Another example, not successful, is pralsetinib in non-small lung. And here, no additional benefit was assigned based on a single arm study and a comparison against a trial arm called properly. Now, the reasoning was comparison not suitable, and that's important for any dossier submission. Methodology needs to be sufficiently documented, and that was not the case here, or at least the GBA came to the conclusion that was not the case here. Second, results were not completely presented. It was mainly on OS and PFS. And then the study pool was considered to be potentially incomplete, and hence, and finally, the observed effect not sufficient to be explained by the intervention, could have been caused by confounding only. Here, obviously, in non-small lung, prazitinib did not have an orphan drug designation and hence was assessed without applying the orphan drug exemptions in the German setting. And finally, a product where the orphan drug exemptions were applicable here, ending up with a non-quantifiable additional benefit based on a single arm and comparing against external control. Reasoning here basically was that in this case, that the indirect comparisons could not be used for the assessment because sufficient information on methodology was simply not there. Now, my conclusion, and I think, yes, my conclusion here would be, yes, external control arms may be a solution on rare occasions. Methodology is difficult. External controls would not help if there are no patient-relevant endpoints in the clinical trials at all, and if trial duration would be considered insufficient. Sample size need to be at least to allow for confounder analysis. And finally, you would need to have dramatic effects in patient-relevant endpoints to be successful in applying external control arms in an assessment. Thank you very much. I think I'm right on time, and I'm happy with all the others to take questions and enter the discussions. There are still a lot of challenges. Thank you very much.
Thank you so much, Mira and Frank Ulrich, for your presentations. Really appreciate it. I understand there are some technical glitches, especially towards the end of my presentation. We'll work to patch those through in the recording that's made available. At this time, I'd like to turn to some of the questions that have come in from the audience in the Q&A. I think, Frank Ulrich, this first one is more directed to you. Could you please give your thoughts on how the use of external control arms is expected to be evaluated within the JCA and whether the level of acceptability may differ across assessors, for example, between Germany and markets that are generally more open to external control data?
Yeah, in principle, I would expect that if Germany, so the ECAs acts as an assessor, they would apply methodological guidance by the implementing acts and guiding documents. So I don't think within the assessment on a European level they would apply, let's say, German rigor. Not to be expected. At the same time, my expectation would be they would clearly distinguish between the joint clinical assessment on the one hand and the German assessment on the other hand. It's not to be expected, at least I would not expect that the quote unquote lenience when it comes to European assessments would be transmitted to German assessments. That's nothing I would expect. Thanks.
Yes, here on the same thing, joint clinical assessment is only presenting data with weaknesses, with strengths, but joint clinical assessment is not concluding, meaning that this is what we have, and each country will decide if it is acceptable or not. However, there is discussion on a disease, on PICO, on what we know, on what we can reasonably think with regards to the data, but there is no conclusion. So that's why it's normally is the play of the game. So I don't know. Anyway, you have here two people, me from France and Frank Ulrich from Germany, and 90% of GCAs are today done by these two countries. So I think we really have to follow the thing. And what is interesting is when you have price taking and price setting countries, when you have price setting countries like France and Germany, there is a possibility to think more about clinical effect, more about unmet need, etc. So it will be interesting. Ask this, I think the same question in 1 year.
Oh, thank you both. The next question that's come through, are there guidelines for ECA use? Are the guidelines for ECA use broadly similar from the EMA and German HTA, or are there challenges found in preference differences from regulator to HTA? And I think that this might be a question that's helpful to have both of your input on. So Mary, maybe from the French perspective, and Frank Ulrich, maybe we can start with you since German system was referenced in the question.
Yeah. So Frank Ulrich, so the only thing is that I didn't succeed of doing for many years is trying to make HTA people do guidelines together. So it was impossible, because every time I said, okay, see, EMA has guidelines, FDA has guidelines, each big country at least has general guidelines on methodology. Nobody wanted to do anything more than that. And every time the answer was general guidelines apply. I really do hope that this will change in 1 or 2 years, because general guidelines do not apply, or with a lot of difficulty when you have gene therapy full stop. So Frank Ulrich, tell me what you think.
Yeah, this actually this reminds me of the discussions prior to implementing joint clinical assessments, and that is the broad variety of methodologies accepted or not accepted across Europe. And my suspicion always was I would not expect that German HTA would adapt or adopt, depends on what we are talking about, would adapt to the methodologies used on a European level. That's nothing I would have expected. This might become more and more challenging over the years, but then we are talking about time horizons 10 years and more, at least from my perspective. So for the time being, I didn't do a comparison on the one hand, the European guidance, and on the right hand side, the German guidance. But there are clear differences, and just to name an example, indirect treatment comparisons will be a matter of fact on the European level, simply because the number of PICOs can't be tackled otherwise. I would not expect acceptance or easy acceptance within the German system. In theory, I briefly presented that ITCs are acceptable. In practice, rejection rate is 90% and higher. So it remains difficult to bridge the gap between the European assessment and the German assessment, and I would not expect a bridge soon. Thanks.
I would be more optimistic than you, because I've spent 3 years working with Beate, working with Stefan Lange, working with people from Belgium, working with others, and we managed to agree on main methodology used for HTA. So I think that the bridge is somewhere here, but at the end, it's acceptability by country that we cannot change. So I think there is a bridge. It will be a bridge, and more now after 1 or 2 years of European assessment, because it is difficult, because you have a lot of open trials, because you have rare cancers. So they will do something, I hope.
Optimism is the privilege of the youth, so that's fine.
Okay.
Thank you very much. Probably have time for 1 or 2 more questions. I think this is one perhaps we can all touch on a little bit, but maybe we can start with you, Mira. How we can increase the acceptability of ECA studies? Any suggestions or ideas that you'd like to raise?
Yes, 2 seconds. Predictable disease evolution and hard endpoints well measured. Full stop.
How about for
And
you,
I would add
Rich?
big effect sizes that cannot be explained by confounding only. And
Yeah.
then, of course, specifically if effect sizes become smaller than dramatic, then a clear description of the methodological approach. That is key.
I agree.
I co-sign onto the responses that you've shared, and I think as was hopefully clear in the portion that I presented, I think really underlying a lot of that is the quality of the data that are being used, and I hope that there are further advances in coming years on this front to better support meaningful RWE generation. So let's think about a final question, and it's a hard choice because there are many great questions in the Q&A that have come through. Let's go with this one. Frank Ulrich, this is another question for you about the German system. Will Germany adapt to accepted EU JCA methodologies, given that the IQWIG is assessing about half of the current assessment cases?
Yeah, and that is a little bit referring back to what I said earlier. Given Mira's optimism, I would say, okay, it might not be 10 years, but perhaps 8. Yeah, so I would not expect an easy and quick adoption of European methodologies. I would expect a lot of resistance, and again, it would be easy if there are dramatic effects. If not, in doubt, I would expect the institutions to stick to well-known methodologies used in the past. That's it.
Again, I'm more optimistic because IQWIG is leading this group in at European Commission. So if IQWIG discusses and finds the common reasoning, I am rather optimistic again.
Thank you. I think with that, we are just at time for the webinar. So thank you both so much for sharing your expertise and experience with us, and for talking us through some of the examples in your home countries. I hope that attendees today have appreciated the content, and that there will be opportunities to continue the conversation on these themes as JCA advances further.
Thank you
Thank you.
so very much, everyone, for joining today, and a huge thank you to our speakers and moderator. As a reminder, the webinar has been recorded and will be made available along with the slides in approximately 2 business days. Genesis will try and reach out via email to answer any outstanding questions not already answered during today's live webinar, but I encourage you to reach out to them at solutions@genesisrg.com so that you can find out more information. Thank you again, everyone.
Bye.
Thank you. Bye-bye.